Hello students, today we are going to learn about the chapter 3 class 12th biology, that is reproduction in human beings. We will start with the human reproductive system, in which first we will start with male reproductive system. So here is the slide which is of a male reproductive system. Here is the overview. You have already studied male reproductive system and its part in 10th standard. So this is just a simple diagram. Now we will start with the labeling of this simple male reproductive system in humans. So one by one we'll start, right? So the first, this is structure which we see is the testis. Okay, now the testis is located inside a pouch which is known as scrotum. Now this scrotum which possess the testis, now outside that testis, the duct which is carrying the sperms which are released by seminiferous tubules. So the next structure coming here is epididymis. So epididymis. Now after epididymis, the duct which is coming out from the scrotum and taking a loop around urinary bladder, this duct is known as vas deferens. When the vas deferens is taking a loop around urinary bladder, it is joining a duct which is coming from the accessory gland, pair of accessory gland, and this accessory gland is actually seminal vesicle. Now the seminal vesicle, when unites with the vas deferens, it forms this part. It forms ejaculatory duct. Okay, now this ejaculatory duct now is uniting with the duct which is coming from, here is the bladder, that is the urinary bladder. So the ejaculatory duct is joining with urinary bladder, and these two ducts when join, here is the entry of single largest accessory gland which is present in human male, that is prostate gland. So here is the prostate gland. Now this prostate gland will join these three, that is ejaculatory duct and the duct which is coming from urinary bladder. It will join from the third accessory gland, that is, we can label here, this gland is Cowper's gland. This is Cowper's gland. Then they will enter into external genitalia, and this external genitalia, it is carrying the urine also and the sperms also. So there is a common opening for, common structure for excretory product and the means the nitrogenous waste, right, and the sperms. So the external genitalia which is carrying the urine and the sperms, that is known as urethra. And the foremost part, the tip of that external genitalia is actually the penis. So the tip of that external genitalia penis is known as glans penis. We will see the detail of this structure again in the coming slide. This is just the overview of male reproductive system, and one by one we will study detailed structures of all the parts. So of course this is actually the abdominal cavity. So here we can write the body cavity. Then here is the ureter, which is coming and joining the urinary bladder. So this labeling is actually for ureter. Now if you have missed something, you can see in the next slide. Yeah, so all the structures we have labeled. So this is the fair out slide. Now we will start with the male reproductive system, that is the detail of different structures present. Actually in males, the testes which are located inside the abdominal cavity, and earlier they are actually intra-abdominal, means they are present inside the abdomen, but at the end of the third month they slowly start descending from the abdomen, and there is a canal which is present. This canal is known as inguinal canal. From this canal the testis starts descending outside the abdominal cavity. Inside that canal is present the fibrous tissue. Look at the fibers, right? So the fibrous tissue which is formed, this fibrous tissue has veins like blood vessels. So blood vessels will carry what? Veins as well as arteries. So veins are present, then arteries are present, then nerve supply is also there. So nerve supply is there, and the nerve supply, the arteries and veins, these all together they are present in the fibrous tissue, and this forms the spermatic cord. It actually forms the cord. Cord means the rope-like structure. So cord, which is spermatic cord, which carries the vast difference which is coming from the testis, blood vessels which has veins as well as arteries, and the nerve supply. So this actually inguinal canal which has a spermatic cord, it attaches the testis with the abdomen, and by the end of the from the till the 7th month, they remain, the testes which are formed, they remain in the inguinal canal, and the 9th month they will completely come out. They will completely become extra abdominal, and this is the characteristic feature of mammalian testes. But some organisms are there like egg-laying mammals, then in even the placental mammals, some animals like bats, elephants, even the marine mammals like dolphins, whales, all they have intra-abdominal testes, because actually the extra-abdominal testes are they are present in the scrotum, which lies outside the abdominal cavity, because the sperms require 2 to 3 degree lower temperature than the normal body temperature. So minus 2 to 3 degree temperature than the lower, and this temperature is provided by scrotum, because scrotum has muscles which are, look at this, cremaster muscle, and one more muscle I'll write here, that is dartos muscle. So the dartos muscles and the cremaster muscle, which are present in the spermatic cord, they help in regulation of temperature inside the scrotum and testes. How? In winters, when the temperature is very cold, okay, so dartos muscles and cremaster muscles, they contract in winters. So they take the scrotum close to the abdominal. So in winter, they will come closer so that they will get enough temperature. In summer, when the body temperature is little bit higher, so what actually happens in wind summers, they relax and they shift the scrotum away from the abdomen. So just close to the abdomen and away from the abdomen, it depends on season. And the dartos muscle and cremaster muscles, they help in regulation of temperature. If it fails, that is the testes fail to descend, okay, then of course there will be no functional maturation of sperms. For maturation, they require 2 to 3 degree lower temperature than the normal body temperature. Rest of the labeling here, which you can see, that is the tunica albuginea layer, tunica vaginalis layer, and the outside the testis, the sperms are carried by the coiled structure, that is epididymis. All these structure we are going to study in the coming slides. Right. Okay. Now, yes, which we have just studied. You can see here what is inguinal canal, the narrow canal which joins the scrotum to the abdomen. This is just a revision. I have already taught you these terms. Inguinal canal. What is spermatic cord, which is made up of fibrous tissue which is present inside the inguinal canal. This spermatic cord has vas deferens, blood vessels, lymph vessels, nerves which are passing, and they are it is joining. Spermatic cord joins all these structures from to scrotum to the abdomen. Okay, and the extra abdominal testes are present in mammals except egg-laying, some other animals like bats, then elephants, then marine mammals like dolphin, whale and all. Okay, now I told you that what is the function of cremaster muscle and dartos muscle, which are present in spermatic cord. Okay, they contract in winters, take the scrotum close to the abdomen. They relax in summers and take scrotum away from the abdomen. Now at last, yeah, if testes fail to descend, then this condition is known as, abnormal condition is known as crypt orchidism. Of course, this will result in infertility. Okay. Now, if single testis is present, that is also an abnormal condition, and the single testis if present, because testis, see the spelling, T-E-S-T-I-S, testis means singular, one testis. Pair of testes are there, pair of testes present. Now if the single testis is present, this condition is known as monorchidism. And natural monorchidism is also present in one animal, that is Ascaris. That's all. Now I'll move to the detailed structure of testis and scrotum. Yeah, different structures present in testicle. Now this is a new term for you, testicle. How will we define this testicle? First we label all the parts and then I'll tell you that what constitute testicle. So this structure we have already done. Okay, that is spermatic cord. No need to explain. Here are the blood vessels, you all know. Now actually this structure is a scrotum plus testis, right? So the lining here, the outer lining, you can see actually these lining, this one and this one. This lining is outermost layer, which is known as tunica vaginalis, and this tunica vaginalis is the covering. It has again different layers, starting from outer layer. Outermost layer is parietal layer, then inner inner to the parietal layer, that is the visceral layer. Okay, now inner to the visceral layer, that is the visceral layer covers the vaginalis cavity. So the vaginalis cavity, that is, you can see here the white line. This line actually covers, look here, this line covers not only the testis, but this line covers the structure which is outside the testis also, outside the, this is actually the scrotum part, and this part is coming out from the scrotum. So this is this structure which is covered by this white line, the complete structure. What is that? That is testis, and forget about the structures which are present in between, but the first structure is testis and the last structure which is epididymis. So this structure is covered by tunica vaginalis cavity, and this structure is known as testicle. So testicle constitute testis as well as epididymis, right? So testicle here is testis plus epididymis, epididymis, right? Now when we going to label all these things, the next layer which is actually the present inside that is the tunica albuginea layer. Now you can see the white fibrous tissue here, right? So this was the vaginalis. This is structure is tunica albuginea. This tunica albuginea is the fibrous layer, and this forms, you can see the white coating inside that also, inside the cavity also. So this tunica vaginalis will move inside, and it will divide the testes into different compartments, and these different compartments are known as lobules. So this structure which is there, it is actually lobule, and you can see this labeling. This one is the septum which is present between different lobules, and this septa are formed by, of course, invagination of that tunica albuginea. Inside that tunica these compartment is the coiled tubules present, and they are known as seminiferous tubule. Seminiferous tubules. Now the compartments which are formed by tunica albuginea, these compartments are known as testicular lobules, and they are around 250 in each testis, and in each lobule 1 to 3, 1, 2 or 3, so 1 to 3 seminiferous tubules which are coiled are present. Actually the main function, the main site of sperm formation is actually the seminiferous tubules. Okay, now this complete structure, because this is actually the scrotum and here is the testis, so you will label here, this is the testis. After the seminiferous tubules, when they release the sperm, these are the coiled part of seminiferous tubules, and here are the straight tubules of coiled tubules, and here are the straight tubules. So this labeling is actually for straight tubules. The sperms which are released from seminiferous tubules, they will come here and they will enter the network. Okay, network of structure here you can see that is actually rete testes. Right? We will see the function of all these structures just now. These after that rete testes, first we label all the parts and then I'll tell you the functions one by one. These rete testes then allow the sperms from rete testes to the coil part epididymis, but before that the structure which joins that rete testis to the coil part, that structure is known as vasa efferentia. Vasa efferentia. Okay. vasa efferentia. Yeah. Now these vasa efferentia actually joined the retitestis with vasa efferentia, right? So this is actually the structure vasa efferentia. Now this structure which is there, the highly coiled, now this structure is epididymis, where the sperms attain functional maturity. When they are released from seminiferous tubules, they are not functionally mature. So the epididymis, which has got three parts, that is the head, this one, okay, and then the body, right, and then the tail part, okay. So this is actually here, you can write here, this is epididymis, which we have labeled here, this is the head, right. Head, you can also write that caput derives from the cap means head, okay. Then the main body part, this one, this is corpus, main body part of the epididymis, and the last part, that is the tail part. So cauda means tail, so cauda. Now here is the tubular part, that's it, nothing else, the coiled part, the duct of epididymis. So the coiled duct of epididymis you can label. When you see the diagram here, yeah, this is the complete diagram which just now we have studied. You can, if you have missed any labeling, you can see here and then label here. So the first was tunica vaginalis, then it forms the tunica vaginalis cavity, which contains seminiferous tubules. These tubules have sperms, because they have the cells which form the sperms, and these sperms will be released from seminiferous tubules to the retitestis. The retitestis will then pass that to the straight tubules, which are vas efferentia. Vas efferentia will then carries and transfer these sperms to the epididymis, and then the epididymis will come out of scrotum and form the vas deferens, which is the main duct which is coming out of scrotum. And we have studied in the previous slide that it is going up in the spermatic cord. It will take a round, a loop around the urinary bladder, and then joins the later accessory ducts and glands. So that's all for the structure. Now we will see, I'll tell you the main functions of the different parts in the next slide. Now again, the first, this is just a detail of that testis, right? So this is actually, we have seen this structure, that is the lobule. So again we can, testicular lobules, right? This is seminiferous tubule, right? Okay, and this one is of course the septa, okay. Then the straight part of seminiferous, this is the coiled part, this is the straight part of seminiferous tubules. So this is also seminiferous tubules, straight part, right, straight tubules. And then it will enter into the retitestis. So 6 is retitestis, and these retitestis will then transfer that sperms to the vas efferentia. These are important one. I'm revising these because the functions of these two here are important one, and then I'll tell you the functions here only, the function of epididymis also. So what is the function of retitestis? Actually, when the sperms are formed in seminiferous tubules, the nourishment is provided, nourishment to the sperms is provided by Sertoli cells. We'll be seeing these structures in the next coming slide. But the sperms are actually present in the liquid which are formed in Sertoli cell, and they are very dilute form. So what is actually the function of retitestis? These are interconnecting tubules, and it is actually the provide to its function is to provide a site for fluid reabsorption. Fluid is reabsorbed in retitestis so that the sperms become concentrated, and then it will actually these retitestis are surrounded by the cuboidal layer which has got microvilli. So cuboidal epithelium with microvilli, and they will guide the sperms to enter into the vas efferentia, and they also help in absorption of liquid. Now the vas efferentia, it is also composed of, it is single layer and composed of columnar ciliated epithelium, and they are of again either they concentrate the liquid part, and also they are meant for absorption of that liquid part. So the sperms which are present in the dilute, which are also known as luminal sperms, right? So luminal sperms get concentrated because of the presence of retitestis and efferent ductules, that is vas efferentia, right? So now we'll come to the very important, the third part. We have seen the function of retitestis, we have seen the function of vas efferentia. Now this is a diagram, and again the labeling. So the first was testicular septa, then seminiferous tubules, then testicular lobules, straight seminiferous tubules, then the vas efferentia, and then the retitestis. Now here the functions of different parts, that is epididymis is a long coiled tube that transports sperm from testis to the vas deferens. Yes, epididymis actually provide functional maturity, because as the sperm passes from the different part of the epididymis, starting from head, then main body, then tail, the sperms get there, the different cells, the number of signals from the epididymis that drive the sperms, and that drive the sperms in their maturation period. There in corpus and corpus, the sperms get the motility movement. They become, motile because it has tail, but they gain motility in the body part, and they can be stored. Functional sperms can be stored in the tail part for few days. So I think it is clear what is the function of retitestis, what is the function of vas efferentia, and what is the function of epididymis. Now the board point of view, this structure is very much important. So we will just see, it is actually what? This is the TS of seminiferous tubule. TS of seminiferous tubule we have just now seen. I'll just draw a rough diagram here. That is the testis is divided into so many compartments, which is known as testicular lobules, and the testicular lobules has 1, 2, 3 seminiferous tubules were there, and these will come into retitestis. So the seminiferous tubules which are present, actually this seminiferous tubule has, this one is single testicular lobule, and here are the seminiferous tubule. So the TS of the seminiferous tubule will give you this structure, the TS of the seminiferous tubule. You can see the seminiferous tubule 1, the seminiferous tubule 2. Here the seminiferous tubule 1. Okay, I'll get the, yes, this seminiferous tubule 1, this one is second, this one is third, like this. So the TS of the seminiferous tubules, and this is the space. Here is the space. This is actually the space between the seminiferous tubules here. This space is the interstitial space. Now we'll see the different structures which is present in the TS of seminiferous tubules. When we see the TS of this seminiferous tubules, of course we'll see the tubule will be round in TS. Okay, so the structures that is present inside, you can see the spermatogonia. Okay, then you can see the different large cells. So this large cell in the tube, these large cells are known as Sertoli cells. First we'll label, and then in the next diagram we will see the functions. First we'll label all the parts. Here the lining of that seminiferous tubule, which is basement membrane. Okay, now here is the germinal epithelium. Children, if you remember the tissue which you have studied, animal tissues in 11th standard, the germinal epithelium is actually the cuboidal epithelium which forms the germs, that is the gametes. And here in the testes, the germinal epithelium undergo meiotic division and it will germinal epithelium. It forms the sperms in different developmental stages. So germinal epithelium will give rise to spermatogonium. Children, one by one you have to see the stages, different developmental stages. You cannot skip a single one because it is actually the different stages, developmental stages here. So germinal epithelium will give spermatogonia. Spermatogonia then will form primary spermatocyte. Of course we'll see this in detail. Primary spermatocyte. Now up here, up to here, only the diploid is there. Now it will undergo the meiosis. So primary will form secondary spermatocyte. Now meiotic 1, meiosis 1 is reductional division. So ploidy will be N, haploidy. Then when the secondary spermatocyte will undergo second meiotic division, it will form 2 spermatid. There is no need to learn right now because all these stages again will come in spermatogenesis. You just have to label it because this diagram will again come. We are just studying here the TS of seminiferous tubule so that we'll be able to see that what type of structures are present inside the seminiferous tubules and what type of structures are present outside the seminiferous tubule, that is in interstitial fluid. Okay, so here are, I've written all the one by one the stages in the development of sperms, and you can see here, you can easily recognize here are the sperms because they have tail. Okay, in interstitial fluid you can see some cells are here, and these cells are Leydig cells or interstitial cells. Leydig cells. Now quickly we will see the functions of these cells. Now here's, you can label, these are the blood vessels, right? I don't think any labeling is left here. So all the labeling you can see in this diagram. Seminiferous tubules, the basement membrane, inside that is the germinal epithelium, then different developmental stages, all the cells are there. Okay, there is 2N, there is 2N, there is 2N, then meiosis 1, N, then meiosis 2 again N, and then transformation of the spermatid to the spermatozoa. Now here the interstitial cells, Leydig cells, which are present in between the seminiferous tubules. Now very important one, that is the different cells which are present inside that seminiferous tubules. So testis, then seminiferous tubules. When we see, then different type of structures present inside the seminiferous tubules. I have just now labeled the diagram. If you recall that diagram, seminiferous tubules inside, there are 2 types of cells inside. What are they? First one, spermatogonia, which are forming the sperm, and here is the Sertoli cell. So 2 types of cells which are present inside, and outside Leydig cells. So I'll write the name of these cells here and then I'll write the functions. Inside the seminiferous tubules, the first cell which is present is Sertoli cells. Children, very very important in the board point of view, because 3-mark question. Draw the TS of seminiferous tubule. Name the cells which are present inside and outside the seminiferous tubules with their functions. Very important general question which is coming. So, Sertoli cell. Now we'll start one by one the function. The first and foremost function is nutrition, nourishment to the developing stages. We have seen that this Sertoli cell is the large cell. Look here. This is actually the large cell present. And this cell is present between the developmental stages of that sperms. And you can also see that sperm head is embedded in the Sertoli cell. Why? Because Sertoli cell is providing nutrition. So the first function is, of course, the nutrition to the sperms. Second one, it secretes androgen binding protein. Androgen binding protein, ABP. I told you that the sperms get the maturation in seminiferous tubules. Outside the seminiferous tubules is epididymis. So due to the influence of that androgen, testosterone, the important androgen is testosterone. So testosterone binding protein, they have the function of concentration of testosterone. Concentration of testosterone. Very important cell. Concentration of, testosterone, which is the male reproductive hormone. And then it is also, it secrete, very important one, another one, that is anti-Mullerian factor. Now what is that? Anti-Mullerian factor, and it is in anti-Mullerian hormone. Actually, Mullerian duct, it gives rise to fallopian tube in female reproductive system. So if the male reproductive system has to develop, then the anti-Mullerian hormone, which is containing anti-Mullerian factor, it has to be secreted so that the development of female reproductive system is stopped. So anti-Mullerian factor, which stops development of female reproductive organ. Development of female reproductive organs, especially fallopian tube and uterus and all, because the Mullerian duct actually associated with the female reproductive structures. So that is why I told you this Sertoli cell, it has very very important functions. Nutrition, besides nutrition, concentration of testosterone and anti-Mullerian factor secretion. The second one we all know, that is spermatogonia, and you know the function. Spermatogonia, you know the function of spermatogonia, that is formation of sperms. The process is known as spermatogenesis. Outside the seminiferous tubules, we have seen the structure Leydig cells, and these Leydig cells are also known as interstitial cell. Why? Because they are present in interstitium. So interstitial cells. Now these interstitial cells secrete androgen, and the important androgen is testosterone, which is the male hormone, and this testosterone will be important for the development of secondary sexual character also, and it has a paracrine effect also, that it helps in spermatogenesis. So that it is important for the practical also, because in your practical the TS of the slide of transfer section of testis is there in your practical exam, and there you will be asked the questions that what are the structures present inside seminiferous tubules and outside seminiferous tubules, and write their functions. So very very important, easy one, but very very important, right? Now here are the chart which we have already formed, which we have already made. These are the different functions of Sertoli cells, spermatogonia, and the interstitial cells. Okay. Yeah. Now the questions. Question number 1. Intra-abdominal testes are found in bat, elephant, whale, and all of the above. See, I told you that generally the testes are extra-abdominal, but in egg-laying mammal and besides egg-laying mammal, some organism like bat, which comes in Chiroptera, elephant, which comes in Proboscidea, then the Cetacea group marine mammal, that is whale and dolphin, the testes are intra-abdominal. Why? Because actually they are these aquatic organisms generally, because they actually have the aquatic environment, and their internal body temperature is optimum for the sperm production. So there is no need for the testes to descend and come outside the abdominal cavity. So what will be the answer here? That is the D. All the above. Now next question we'll see. Vas efferentia connect. If you remember, there was a diagram, that is the testicular lobules were there. After there, then the retitestis, the coiled structures were there. After that, the straight tubules, and the straight tubules that lead to the structure which is highly coiled and has got the 3 parts. If you remember, children, these are testicular lobules. This structure is retitestis. The straight tubules is actually the vas efferentia, right? And this one is epididymis. So the vas efferentia has the function of joining retitestis to epididymis. So what will be the answer? Vas efferentia connect. Testis with epididymis? No. Kidneys with epididymis? No. Testes with urinogenital duct? And none of the above. What will be the answer? Yes, see, when we see that testis with epididymis, because the retitestis is also there, but before the retitestis is the testicular lobule. So here the most prominent answer we can write that testis with epididymis. So A answer. Actually the correct answer will be, the more correct answer will be just now testis but the retitestis. But if it's written here testis, so it is also joining indirectly testis to the epididymis. So the answer will be A. Okay? Yeah. The next question. Yeah. Fill in the blanks. Here seminiferous tubules, retitestis. Just now we have seen which structure? Vas efferentia. Okay. Epididymis. This epididymis come out from scrotum and joins what? Joins vas deferens. This vas deferens, if you remember children, this vas deferens taking a loop around urinary bladder and then joining the duct which is coming out from seminal vesicle, and then it will enter into the external genitalia that is containing urethra, because it is coming from urinary bladder. Okay. Yeah, this is the answer, right? Now we'll see the next question. State the role of epididymis in male fertility. I told you that epididymis has different part, and when the sperms are released from seminiferous tubules, it will be carried to epididymis by retitestis and then vas efferentia, so that they will attain sexual maturity. Because epididymis is related with the sexual and functional maturity of sperms, so it is also related with the male fertility. Okay, So the functional maturity is gained in epididymis because the surface of the sperm is altered in response to the secretions of the epididymis. And this is actually important. Why? Because that response which is present on the surface of the sperms, we will see at the time of fertilization there. So the conditioning of the sperms occur when it enters inside the egg, and that conditioning will enable the sperm to fertilize the egg. That functional maturity is given to the sperm by the secretion of epididymis. Okay, so the answer is, yeah, spermatozoa acquired motility and capacity to fertilize egg. That is very, very important. 2 points you have to write. So that's all, children. For today, we have studied the various structures present in human male reproductive system and their functions. Thank you.